Hello, everyone, and welcome to today's keynote presentation, Measurements and Standards to Support Confidence in Genome Editing, presented by Dr. Samantha Maragh, leader of the Genome Editing Program at the National Institute of Standards and Technology. I'm Dr. Susie Valdez of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I want to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on that Ask a Question box on the far left of your screen and type your questions into the drop-down box that appear on the screen. Our speaker will respond to your questions via email. If you have any trouble seeing or hearing this presentation, just click on that Ask a Question box and let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Just click on that Continuing Education Credits tab located at the top right-hand corner of your presentation window and follow the process to obtain those credits. Without further ado, please join me in welcoming our keynote presenter, Dr. Samantha Maragh. I will now turn the presentation over to her. Welcome, Dr. Maragh.
Thank you so much for having me. I'm so glad to be here to present at this inaugural LabRoots meeting on CRISPR technologies. So my name is Samantha Maragh. I'm from the U.S. National Institute of Standards and Technology. And many of you may not be familiar with us, but I'm going to familiarize you with who we are, what we do, and how we're impacting the field of genome editing. So the talk title is Measurements and Standards to Support Confidence in Genome Editing. So first, I'll go over the talk and the outline for today. So I'm going to give you an introduction to NIST and what measurement assurance is. This may be a title and term that you're not familiar with. So what exactly that means and how it's important and impacts every aspect of the science that you do. Next, I'll go into the measurement challenges for genome editing as a field, where CRISPR is one of the leading technologies. And then I'm going to introduce you to the NIST Genome Editing Consortium, and that is an effort started here at NIST to support the genome editing community and translation of this technology space into industry and potentially products that can impact our everyday life. So we'll begin with an introduction to NIST. So NIST, who are we? NIST, as I mentioned, is the U.S. National Metrology Institute. So our goal is global harmonization of measurements. It sounds like it might not really fit into the thought process of where biology is, but everything around you, everything you do is based on some measurement. Time, weight, how much volume is in your pipetter. All of these things are measurements. And NIST, we're the people that help make sure that you understand whether those measurements are correct. We also consider ourselves industries lab. So NIST is part of the Department of Commerce. So we're not part of Health and Human Services like NIH or FDA. We're part of a different part of the government. And our job is to help U.S. competitiveness and translation of technologies. So our mission is really in the cutting edge of technology, measurements, and standards. When you go to the doctor and they take your cholesterol, how do you know that that answer is right? We're the people that are working to help make sure that there's confidence in those kinds of measurements, as well as other kinds of measurements. And technology. So technology development and transfer is something that we also do. So how can we help a new technology space, like the genome editing field, develop tools that meet new kinds of measurement capabilities? Biology. So if you are familiar with NIST, you may know of us as the people that make the atomic clock that time is calibrated to, or weights and measures. But NIST is also very focused and engaged in the biology sector. And so that means biology that has to do with pharmaceuticals, biology that might have to do with food, or other sorts of health or environment are the kinds of things that we also support. So we work very closely with other government agencies, with industry, with those that are at the cutting edge of research development in order to help translate these technologies and these tools. And so NIST, we're measurement people. So we draw from our broad range of expertise in measurement science to apply to the new and growing types of challenges. And we are the trusted party. So NIST, we're not regulatory. We don't enforce anything on people, but we are there to partner. So we're a trusted third party. We are meant to be unbiased and provide that level of collaboration in this sector. So now that I've given you a little bit of an introduction to NIST, I want to give you an introduction to why and how NIST is interacting in the biology space. So there's been, over the last several years, this recognition of this concept of irreproducibility. There have been several studies, and I'm just highlighting one here, into the economics of this, how much money is going into the biological sciences and how much money maybe isn't being used effectively because of irreproducibility. And so there is a burden on our research environment of data that's not reproducible. But the question really is that the goal? Is the goal to be able to get the same number over and over again? So I would suggest that the answer to that is no. You can reproduce a number, but that doesn't mean that you're confident that the number is right. For example, you could set your clock 5 minutes ahead because you want to make sure that you arrive places on time. Every time you look at your clock at 7 in the morning, it tells you that it's 6:55. You can do that every day, day in and day out. Somebody else can look at your clock, but that doesn't mean that you have confidence that it is actually 7 o'clock. So that's the kind of thing that we're talking about. These things can be systematic biases, or there can be things in your system that you can reproduce the same number every time, and it still might not be the right thing. So the idea is we should go beyond reproducibility. We should really understand the measurements that we're making and what might cause them to be impacted. So we need to understand what it is that we're trying to measure, how the process works to measure it, what are the ways that that measurement can be impacted, to assess whether we're really confident in the data that we have and that we trust it to make important decisions. So if we dig into these concepts that go beyond reproducibility, what do they look like? So why might you get irreproducibility? It might be a system of a broader challenge in biomedical research. So irreproducibility, it might not actually be your enemy. It might be telling you something about what's actually happening. Maybe you don't get the same result because this lot of your reagent is actually biologically different than a new lot of reagent. Maybe this lot of cells is biologically different than another lot of cells. And it's telling you that something real is happening because biology is very complex. And the systems that we use to measure biology can be very complex. From the NIST perspective, what you really need is really rigorous ways of assessing your process and understanding whether your process is trustworthy. And so why and how might you be able to get some of that confidence, understand whether the answer is right? That comes in having some control materials or some statistical tools or maybe some reference data that can help you understand whether the answer that you got on a particular day, maybe at a particular time, maybe a particular operator, is trustworthy, and if it's reasonable to expect that result. And what you really need is it requires some initial work, some initial testing to say what is the right range to expect. And so those sorts of biological tools which lead to greater confidence are sometimes the tools that NIST helps develop or helps a community implement. So if you don't really remember any of that, and if you are still struggling with this idea of confidence in your data, this is really the one slide to really take away from this conversation right now. It's that the sources of pre-analytical and analytical variability, they have to be accounted for to tease out true biological signal. So what we're really saying here is that at the end of a process, you may run a sample and you may get a positive on your sample, whatever positive means for your system. And you may say, there's a difference between this sample that I got a positive on and this other sample that you have a negative on. And you want to be able to move forward with that understanding. But why are you getting a positive signal? Is it really the biology, or is it because these two samples are run on 2 different days, and on one day, the instrument wasn't working the same way, or they were run by 2 different operators, and one of the operators gets a different answer than the other? So you really need to understand what are the ways that your result might be impacted by the different factors that are part of your measurement process to be able to say, I believe that this result is attributable to the biology that I'm trying to measure, as opposed to these assay or these instrument variables or these human variables that are causing that result. So then how do we even go about this process? What makes good data? So what makes good data? We've formulated a list that gives some understanding of these principles of metrology or measurement science. So sometimes these terms are used interchangeably, but they really do have discrete and different definitions that can help you understand your data better. So this idea of precision, so replicates and reproducibility within a certain context. You can have that. You can have really good precision, but you don't necessarily have accuracy. The result might not actually be true to the expected that you would see. And that's where if you have different kinds of measurements trying to measure the same kind of thing, then that can get you at some accuracy. What is actually the correct value? How close are you to the correct value? Robustness. So if you change, if you are meant to pipette 10 microliters, if you pipette 10.1, does that matter? If you pipette 11, does that matter? How robust is your system? Does it matter if you use a different instrument? Let's say you're doing PCR. Does it matter which PCR instrument you use? How robust is your process? That's what we're talking about here. Now, the knowledge of the limits of detection of your assay is very important. There are often times that you get data that's reproducible, but it's outside the dynamic range of your assay, which really means your assay can't give you reliable information in that space that you're getting signal. And so you may get that signal reproducibly, but it's not actually the value that you're measuring that you can trust. So this often happens with assays when we don't characterize the limits of detection, and we make the mistake of using values that are outside the dynamic range. Now, this is a big one for NIST, the use of benchmark materials. So one of the things that NIST does is generate benchmark materials that can help a community understand what their measurement pipeline is giving them. So sometimes we call these things truth in a bottle, something that has some value that's certified that lets you understand whether in your hands, in your process, you're getting the right value because we've given you a value that you should expect to get. And very importantly, and something that we can all do better at, I feel, is reporting. So we often do very important things and very sophisticated things and do very good and thorough science. And sometimes it gets lost in the reporting process. And data and the metadata, how you arrived at your answer, are really critical to understanding a measurement process and understanding the quality of your data, because it may be in the details and not the final answer that's what's causing a lack of comparability or reproducibility when one should expect to get reproducibility. So I just mentioned reference materials. So let me tell you about the kinds of things that NIST generates. I've given you some concept for the kinds of challenges that you deal with every day in your research that we might be able to help with what NIST does here. But these are the physical kinds of things that NIST actually delivers to people. Documentary standards. So these are norms or protocols that are able to help come to some normalization within a field. Reference materials, I just mentioned this. These are physical things that you can get in your hands that have some certified value so that you know in your hands whether you're getting the right answer. and reference data. So these are data sets, databases, where the data that goes along with some of these reference materials or these reference protocols that you can get in your hands and for your informatics and your bioinformatics pipelines and processes, you can see if those sorts of tools are giving you reliable outputs. So how are these things all built up? So I talk about NIST. I tell you that we're not a regulatory agency. We don't enforce standards. Standards are always adopted by consensus by experts in the community. So we work collaboratively. And it begins with assays. So what are the assays that we want to understand? Then it goes on to the measurement assurance. So what are the ways that we use to understand how these assays perform? So we use things like interlab studies, where we send a sample to multiple labs and see what answers people get and maybe why those answers might be different. As well as design of experiments, taking an assay and saying, well, where are all the places that are really critical that if you change something here, you might get a different answer? And then eventually, if it's the right kind of output to be developed, that might result in guidance or methods that are normalized. Here's one real example that I want to give you of work, something that NIST has done and developed that's really relevant to the genome editing space and the CRISPR field as it's developing and becoming more mature. So this Genome in a Bottle Consortium is a NIST-led effort to address the issue. It started several years ago, around 2012, to address the issue of next-generation sequencing. So this was this shift in technology, which we've all come to recognize as a very powerful resource that's pretty much a part of most people's pipelines right now. And that is a powerful tool that wanted to have an application in the clinic. And one of the challenges with a new technology is, how do you know if you can trust it, especially to use for very important kinds of applications? And so this was where the next generation sequencing toolmakers wanted to be able to use these devices as clinical diagnostic devices. So to be able to diagnose genetic disorders and things of that nature, or to be able to screen for genetic variants. And this was one of those things, well, how do you know if these sorts of tools are supposed to be able to find the unknown unknown? The next generation sequencers, you put whatever in and you should figure out what its sequence is. Then how would the FDA know if the information was true? You're giving back sequence out of the NGS sequencer, but is it biologically associated with the sample you actually put in the process? So NIST worked with a wide group of collaborators and experts in the DNA sequencing space and the bioinformatics space to develop this material. And it is the single most highly characterized human genome set of materials that are out there. And these materials were used to get the first genome sequencer into the clinic for NGS sequencing to be a clinical device. And so we partnered with FDA as NIST and FDA to be able to address this challenge. And so this material is useful for anybody who wants to understand how variable is their general genome editing process. It's purified DNA. The NIST has certified every base within this human genome. So if we say that there's an A at this position, we say it's an A, and here's our confidence that it actually is an A. And we've collaborated widely to make sure that this material is really fit for the purpose that it was designed for. And so this is something that allows you, there's this stoplight here. And the stoplight is, we refer to these as yellow light standards. If you get the sample in your hands, you run it through your NGS process, and you get the correct information. And by correct, I'm saying the information that is consistent with the certified values, then that's a yellow light. That's a proceed with caution. Your pipeline seems to be working well. If you get a red light, that's a stop. Something went wrong, reevaluate your process. Important thing to know is that this doesn't tell you about maybe the disease variant that you care about, but it tells you about your ability to generally get reliable information out of your NGS sequencing pipeline. Here's some additional information. These are the samples that are available as the Genome in a Bottle certified DNA samples. And here is the vast number of NGS sequencing platforms that the samples have been analyzed on. The cell lines for these samples are available through Coriell. And if you'd like to get any of the certified DNA, that is available through NIST. One very interesting use, I mentioned standard data before, is how FDA has used this collaboration as this Precision FDA Truth Challenge. So these Genome in a Bottle samples, there are now 7 total. They were released not all at the same time. And at one point, FDA had NIST hold back the data for one of the Genome in a Bottle samples. Along with the DNA, we released all of the data that came from those NGS analyses. And so they did this Precision FDA Truth Challenge. So what this is it allowed the user community to take the Genome in a Bottle DNA sample, do the sequencing, use their own tools to do de novo assembly of the genome, and then that was submitted to FDA. That was then given to NIST, and we assessed it to see which assembler, which pipeline gave the results that were most similar to the certified values. And this was another unique way to use the kind of outputs that NIST generates to be able to help the community in general. And then out of this, some new pipelines for de novo assembly were able to be confirmed and validated. So just to finish off with NIST, what does NIST do and how does NIST work with communities? NIST works internationally. We work with other government, industry, academia. We work nationally, internationally. So this really covers basically anybody in a sector NIST is able to work with and strives to work with in order to create harmonization in an area. One unique sort of interaction I'll mention is NIST's relationship with the FDA, which we collaborate with closely. NIST as measurement experts, FDA as regulators who are able to see pre-competitive needs that, man, if they had something that some standard or some data that would help a field, they could see how it might be able to accelerate a technology space to be able to develop therapies that could help people, or new tools that could help people, or get new kinds of products into the market. And we work regularly with them in order to be able to do that and support them as another government agency with our expertise. So now that I've given you an overview of NIST, let's dive into the measurement challenges, the thing that you're really here for, which is the genome editing and the CRISPR technologies. So how does this really apply to NIST and the work that we're doing? Well, let me begin with the scope of what we're talking about, targeted genome editing. And so if we describe that roughly as the capability to make targeted DNA sequence changes in live cells, then that's being aggressively pursued for bioscience applications. So all sorts of applications, from basic research to material sciences to health sciences and agriculture. So this is a sector that really fits well with something that NIST sees as being able to have a wide and very deep impact on the economy and technology. So were there needs that NIST could help with? So the answer came to us very quickly as yes, there were applications that there were needs, and these applications were particularly high in their need for having confidence in the kinds of measurements. And the area that came most strongly forward was those that are trying to make therapies based on genome editing. So those that are trying to make gene therapies particularly. So the idea is that there are industry members that are trying to take cells from patients, be able to use genome editing, CRISPR, and other tools to engineer these cells, and to be able to deliver them back to a patient as a therapeutic. And in addition to this mode, that people are trying to potentially formulate these genome editing molecules to be able to say, deliver CRISPR directly into a person. And so the idea of doing that in a new technology, you really need to have very rigorous measurement science to make sure that you feel comfortable with being able to translate this kind of technology into the clinic. If we look at a very general genome editing overview, then we can bin this into a few very large bins. So let's look at this process. So let's say you start with genome editing molecules. And at the beginning, there are very many kinds of editing molecules. This meeting is focused on CRISPR, but the kinds of tools and things that NIST generates would be applicable to CRISPR as well as other tools, including zinc fingers, talons, and the derivatives of CRISPR like base editors. So once you decide on your target and what kind of molecule you're going to use, you have to figure out how you're going to formulate it. Are you going to deliver this molecule to cells as DNA, as RNA, as protein? What's possible? What kind of molecules are you delivering? And then how are you going to get them into the cell? Are you going to do some sort of electroporation? Are you going to use some sort of virus? Is it lentivirus? Is it adenovirus? Is it a nanoparticle? What is it that you're using? Then you've got to, hopefully, your molecule works and you get the mechanism of action. It's a strand break, single strand, double strand break, it's a nick, whatever that mechanism of action is. And then hopefully, if your processes are working, then you get an edit generated as a result of this DNA perturbation. And then you need to confirm what you did. And then you've got to determine the fit for purpose. So even after you confirm what you did, is this product fit for the purpose that you need it for? All along this set of places in your process are places where there can be variability. You can make different choices, and depending on the choices you make, that may impact your output. And this is really where we're helping the community understand the variability in the process and understand, given the measurements that they're trying to use, how confident they can be in them. So let's dig a little bit more into this idea of a gene therapy. So you see here where we start with cells coming out of a patient, then this genome editing process and what you might get. You might get unedited cells, you might get cells that have your desired edits, and you might have cells that have your undesired edits. Both the cells that have your desired edits, they may also have undesired edits in addition to your desired edits. So this is a complex kind of pool that could come out of a genome editing process. And to add to that sort of complexity of the heterogeneity of what you might get at the population level, when we dig into that a little bit more and look at the DNA level, how complex might this be? Well, it can be very complex. So each cell is their own little bioreactor, you can consider it. And what happens in one cell doesn't determine what's going to happen in another cell. So the events that are happening where your genome editing molecules are interacting with your DNA and how many locations they interact with your DNA, this can be your on target, the edit location you meant for them to act at, and these off-target locations in the genome you didn't mean for them to do anything at. So those vary between cells because they're completely independent events. And what comes out of this process can be very complex. So because they're independent events, the repair outcomes within each cell can vary. And because these analyses are done mostly not at the single cell level, it can be very difficult to do that and expensive to do. You're pooling cells and doing a sequencing, and you may get something that looks like what we see in the second step here, which is a lot of variants at the same location and different kinds of variants that are generated. Insertions, deletions, maybe single base pair substitutions, and the size of these insertions and deletions can be larger or smaller. So the ability to detect this and for your bioinformatics to report this out appropriately, this can be variable, and it's a challenge to understand. NIST recognized this, but we weren't really sure if there was truly a need in this space. So we hosted a workshop in 2016, and the output of that was that, yes, we had very strong consensus that there was a need for NIST in this space, and that there were ways for the community to work together in this space. So the things that came out of this were confidence in existing assays, measurements that you're already using to assess what happened in your genome editing experiments, that there should be a little bit more confidence there. informatics and data confidence norms that, man, it would be really great if there was some way of harmonizing, normalizing, and understanding how variable the data and the informatics tools to generate that data or analyze that data are, and that there's the space and opportunity for new assays to meet unmet measurement capability. We followed up with the field, and here's a list. I'm not going to read it because you can see it there, of questions that came out that were consistent across many different leaders in the field that were places that NIST might be able to help the community. So what came out of all of these discussions, and here in the box that has the company logos, if a company is in that box, at some point they participated in the workshops or the discussions that NIST hosted to understand what the needs were. So what came out of this was the need for a NIST consortium to help in this sector. And there were 3 target areas that were identified: specificity measurements, data and metadata, and lexicon, which I hadn't discussed yet, and I'll discuss a little bit more later. So I'm going to move on to the last part of the talk, which is talking about the NIST Genome Editing Consortium. And I will go through the general parts of the consortium and the work that we're building right now. So the NIST Genome Editing Consortium, this consortium is active as of June 1st, 2018. And the goal of this consortium is to convene experts across every sector of the genome editing field to address the measurements and standards needs to increase the confidence of using genome editing and the translation of these tools in research and commercial products. So I've got this diagram again that we saw earlier, and really where we're acting at is understanding what assays we need to work with and giving measurement assurance for assays, data, all parts of this process. So there are some consortium goals. The goals include, on the assay side, qualifying the assays to evaluate the genome editing outputs. These might be assays for on-target and off-target editing. They might be assays that you use to assess where in the genome an editing molecule might have acted. To develop physical reference materials that allow you to have something in a bottle, something in a vial in your own hands that allows you to say, how well is this assay doing in my hands? To generate reference data, so data and informatics pipelines, the data that might go along with that physical sample, to say, how well is my informatics pipeline doing at getting the similar result that is generated out of this consortium? And suggested minimum information guidelines. So when you do a genome editing study, when you're doing your CRISPR experiments, when you go to publish or when you go to post that information, what information should we always be thinking about putting and keeping with that to help the next person that's using that data, using that information, know exactly what was done? And lastly, generate a common lexicon for genome editing studies. So this is terminology. How are words used? When are they used? Do they mean the same thing? Is gene editing the same as genome editing, the same as gene or genome engineering? Is a guide RNA the same as a single guide RNA? Is it the same as as the tracer plus CRISPR RNA? What are we talking about? And let's have some normalization, which will accelerate and forward the commutability of studies. So our goal at the end of this consortium is to have shared materials that can accelerate the field adoption and translation of genome editing technologies to products and other sectors, norms that allow for greater understanding and forwarding the field of genome editing at an even more rapid rate, improved understanding of our confidence in detecting on-target and particularly off-target genome editing. So you know where it is, you know what it is, and then you can begin to say, well, what might it be doing, or does it really matter? And then harmonize genome editing so that we can all be speaking the same language and move forward together. So the things that I just talked about, the goals of the consortium, they are divided into 3 working groups. One is roughly the working group that we might be able to call wet lab. We call it specificity measurements. The second working group is the data and metadata working group. And the third working group is the lexicon working group. And I'm going to go over each and every one of these in a little bit more detail. So what I want to share with you is our current membership of the consortium. So this slide just lists our current members. We're still accepting members to the consortium, and members will be accepted on a rolling basis. And members can participate in all or even just one aspect of the consortium as they see their expertise fits. So working group 1 of the consortium, specificity measurements. So specificity is meant to be assay qualification, which is a term that I mentioned but didn't really describe, and also development of reference materials, which I've gone over a little bit more. So assay qualification process, what does this really mean? It really means for you to understand how good your assay is. So if I hearken back to that list earlier of, well, how good is your measurement? That's what we're trying to understand when we do a assay qualification process. At the end of this, I have these steps here that you can read. At the end of this, you want to have an understanding of the precision of an assay, the robustness of an assay, the dynamic range of an assay, and all of this should be put along with the metadata so people understand how you got to those values. So for this consortium, we'll first need to identify what are the core and the most important assays for NIST to evaluate. And then we'll be able to go through this process with the consortium members to develop what should be an assessment of these critical assays that have utility for especially understanding the clinical applications of genome editing and CRISPR technologies. So understanding the assay dynamic range requires the ability to change variables and assess how that change impacted your output. You can refine the assays and protocols, establish what the baseline is. So what if you just ran an assay, but you didn't try to do the editing? How variable is just running an assay without trying to use CRISPR? That's very important to know what your baseline is. So when you get a result, you can say, is this biologically relevant, or is just the noise of happening to run this assay? And then after you understand and you've qualified the assay, you can better understand what you should come out of this, what control can come out of this. I've discussed reference materials. I'm going to put this up here. One of the important things about reference materials is to specify what the material needs to do for you. It's not going to do everything. It's not meant to do everything, but what is it actually going to do for you? And design it to do that, and then test its ability to do that. And for this consortium, we're looking at DNA-based reference materials. So it won't control for the editing process itself. It won't control for DNA extraction, but it would control for after you have an edited sample and you want to assess it, this is where you could carry this forward and use it to assess the rest of your process and the rest of your assays you use. So one of the ways, this genome in a bottle reference material that I mentioned earlier, it's very useful and helpful for understanding general genome editing, but something that looks like, it's helpful to understand general NGS sequencing. But for something that looks like genome editing, you're going to need something that has the complexity that looks like a genome edited sample. So here's, again, another way of looking at the simplified genome editing process. And I just mentioned the assay qualification and the reference materials. And at the point where you might screen for genomic disruption, now you're not necessarily saying what happened, but where something happened. Assay qualification can be helpful because these assays, many of them are newer and haven't been qualified the way that, say, NGS has been qualified. And where you're using some of these sequencing types of assays for the genome editing application, a reference material that gives you something that looks like a genome edited sample is the kind of fit that we're working towards. So just briefly, I'm going to round out here with what is this kind of thing that NIST might generate. So this was an original idea where we would have a DNA sample we might spike in variants that look like genome editing, and then we might QC that and maybe say, okay, well, we've got all of these different genome editing variants. What mixture should we use? Should it be 10% of this A and then 2% of this C and then 1% of this 2 base pair deletion? What does that look like? So this is a process that we're thinking about implementing, and we got some feedback. We thought about it a little bit more, and this isn't really the actual fit for purpose that we're going to go forward with. The idea of a spike in, it doesn't really suit all of the many ways that a genome editing sample might be used and the kinds of assays people might want to use it for. So engineering in variants to make a control seems like a much better process that will allow you to have some more control and have variants in context that might be more valuable than a short oligo that's spiked into a large batch of DNA. So we're refining the thought process after some additional thought and input from experts as to what really would be valuable for them is engineering in variants into cells and growing up single cell clonal populations, extracting DNA, measuring the DNA that come from the independent single cell clonal populations, and then determining what mixture would be helpful as a mimic of on-target and off-target DNA editing. So the outputs from this would be a series of DNA process control materials, and mixed samples as well as unmixed samples will be provided. We would QC these samples with qPCR and NGS sequencing, and those assays and the data from those assays would be an output of this, as well as the data and all of the metadata associated with this as an output and a tool to be used by the community. So briefly, I'm just going to mention Working Group 2 and Working Group 3. Working Group 2 is data and metadata. So data and metadata, simply there is a need to both capture information, have ways of sharing the information and comparing the information. And so this comes in really two flavors for what this working group's gonna do, which is community norms for minimum information reporting, which I've mentioned earlier. What are these terms? How would they be housed? How would we share this data and metadata? And community norms for data formats and tools for benchmarking analysis. What is a minimum norm for a data file format so that different kinds of informatics tools can use the same file to be able to process? And there are different informatics tools. Is there some set of norms to understand how to compare outputs from different bioinformatics tools? Lastly, the lexicon working group. So the lexicon working group is meant to come to some minimum norms on terminology. And so if we have that, it should enable clear communication of scientific results, facilitate communication within the expert community and with regulators like FDA and the USDA, and have the potential for international acceptance as international terminology norms. So right now we have this pilot list of terms that we are trying to narrow down a first list of terms to begin to define minimum definitions. And so we're looking for a high priority list of maybe about 20 terms that are used differentially or used in ambiguous ways or ways that are inconsistent either within any particular subfield or across different fields, maybe between human applications and non-human applications, and begin to generate a pilot definition that we can share widely with the community. Here's a link on this slide that you can go to that website and give information about this minimum list that we're gathering. So lastly, there was, and we do, share and interact widely through workshops. And there was recently a NIST FDA workshop to basically share FDA's perspective on what they might see as needs if you're wanting to use CRISPR genome editing for regulated purposes and develop therapies. And through this, we were able to clarify regulatory perspectives. And what I just shared with you all, I shared at this workshop and got some of the refinement and additional input. And we were able to identify and get some more input for a path forward. So workshops are another way that we interact with the community, and I welcome you to participate in future NIST workshops. So lastly, the path for success. So NIST trying to help the genome editing field to impact and create greater outputs for the science that you're doing in the CRISPR field. This is the path for success. We want to continue signing up consortium participants, including international and within the US of different expertises, to figure out their most effective contributions, identify the experts that can contribute to the reference material development and analysis, as well as the data and metadata, and to be able to integrate all of these activities across what's being developed in the experimental wet lab side with the data and informatics and the terminology experts. So thank you so much for participating in this talk, and I look forward to helping you as NIST.
And thank you, Dr. Maragh, for that informative presentation. We will now start the Q&A portion of our webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question drop-down box located on the far left of your presentation window. Type your questions into that box that appear on the screen and click the send button. Our speaker will follow up with your questions via email. So let's take a look at some of the questions from our viewers. Dr. Maragh, does everyone need to use standards, or is this just for the industry?
So in terms of standards, I think it depends on what you're doing, what kind of confidence tool you need. But as scientists, we should be introducing some sorts of standards into our work. And in this aspect, the kinds of things that you need to do, this idea of fit for purpose. What are you doing and what is your fit for purpose? And if you want to be able to trust your data, you need to implement some sort of standard or norm, whether it's within your own hands, but shared norms are definitely an important thing. And I would hope that the audience would be interested in engaging in defining what it is that you really need, because these things should be developed by consensus in order to fit a need that you have.
Thank you. And will any of these standards be required for publication?
So we don't require anything as NIST, but we have had interactions with some journals that are very interested in the idea of minimum information norms. They are a regular part of other aspects of other fields. NIST has been involved with qPCR and digital PCR minimum information norms, and those have been adopted as expected norms that people would follow when they publish on those sorts of data. So it may be something that becomes part of the publication infrastructure, although NIST wouldn't be the one that implements anything like that.
And Dr. Maragh, what about CRISPR for other applications besides medicine?
So that's a very important and very valuable field. The field that we've been focusing on for this initial consortium work has been the gene therapy field. It's been the one that had really come forward the most and said that they had a really pressing need, and that was consistent with what we were hearing from FDA. But a lot of the tools that we're developing are really fundamental capabilities. So your ability to understand if you have a genome edited sample, and how good is your NGS in your hands to detect things that look like a genome edited sample. There may be some differences between applications, but that's a core competency. So we're trying to get down to core competencies. Data, metadata, even though our use case is gene therapy for the most part, that's a core capability that goes beyond just medical applications of these tools. And certainly terminology. Our goal is to make sure that the minimum terminology is applicable to all fields and not just for a medical use of that term.
Dr. Maragh, thank you for being our keynote presenter today. Do you have any closing remarks you'd like to provide our viewers?
I appreciate you having me today. I encourage all of you, NIST is here to collaborate with and to help the community at large. I encourage you to get in touch with me. I'm leading the genome editing program as well as the consortium if you have any questions. But NIST is here to do things to accelerate this field, and I just look forward to working with this community more in the future.
Well, thank you as well, and thank you again for your important research. I'd also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to remind everyone that today's webcast will be available for on-demand viewing through January 2019. You'll receive an email from LabRoots letting you know that the webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now, and thank you for joining us. We hope to see you again soon. Bye-bye.